to see that one.
Thank you for the great help. Thank you for the great help and for the consent to address us this afternoon.
Yeah, yes, that is not an issue because as Dr. Sujit has mentioned, for me, he is my like a brother. We in Calicut University, we nearly shared our, because actually I moved from my room to Dr. Sujit's room for more than 1.5 years. So we were sharing everything. I mean, sharing room, because they were great seniors for us, because they were guiding us. They were giving us proper directions, and we still, that is one of the best period in my life. So whenever he contact, I don't need to have any kind of a second thought to accept his invitation.
Right.
And also I would like to discuss something about the latest development of supramolecular chemistry and its relationship with our life, as well as some applications. So I just told him that one, okay, I will give a talk. It's something different. It is something more contemporary because of this. Because I was hearing last week Professor Jean-Marie Lyon's lecture. So he was explaining us that one, what is its importance of supramolecular chemistry. So taking inspiration from that one, I would like to share some of ideas about the latest development in supramolecular chemistry.
Wonderful. Sujit, can we start now?
Yes, sir, we can start now. Sandhyarani, can you hear me?
Yes, Sujit.
Yes, then we can start. Okay.
Yeah.
So, welcome all of you. So, I'm not taking much time because we are late. As I told you, Dr. Praveen and we were together in Calicut University. So I know that he is a very hardworking person, and he has also a very good passion to the subject. So that actually inspired all of us at that time. Okay, and he is also a man with a very good social commitment. And another person is our beloved Sandhyarani madam. She is going to chair the session. Let me say a few words about Professor Sandhyarani. Dr. Sandhyarani is presently a professor at School of Material Science and Engineering. And having completed MSc Chemistry from School of Chemical Sciences, MG University, she earned a PhD degree in chemistry working with Professor T. Pradeep at the Indian Institute of Technology, Madras. Subsequently, she joined Pohang University of Science and Technology, South Korea, as a visiting scholar in 2000. Later, she spent about as postdoctoral fellow at University of Notre Dame, and also in Cornell University, Ithaca, New York. She then joined BITS Pilani Goa campus as lecturer and worked there in 2006. In 2007, she joined NIT Calicut as an assistant professor, where she is a full professor now. She published around 60 articles in international journals with an item index of 53 and an H index of 26. She has taken 6 sponsored projects of PhD, PBT, CSIR, and CASTEC. 6 PhD students are completed their PhD under her guidance, and 6 students are presently pursuing their PhD with her. Her research interests include the application of nanomaterials in clean energy and healthcare. And the research group particularly focus on development of electrocatalysts for methanol oxidation and oxygen reduction reactions for the application of fuel cells. And also, she is working on photocatalytic application, water splitting, energy storage devices, fuel cell and biosensors, DNA sensors, dopamine sensors, and endotoxin sensors. So I welcome both of you, both the personalities to this session, and I welcome all of you for the session. And now I invite Professor Sandhyarani to chair the session and to introduce Dr. Praveen. Thank you.
Yeah, thank you, Sujit. Actually, I'm not able to switch on my video. There is some background noise coming in. If all others could mute it, it would be good.
So, Juraj, can you mute the mic?
Switch from Juraj.
Juraj, can I mute?
Yeah, maybe Sujit, if you can do like mute all the participants from this and then allow only person who are speaking, that would be better.
Okay, so now I am muting. Okay.
Yeah, you can mute everyone.
Muting all means you are
No,
you
then
also will be muted.
yeah, just mute all. Then you.
Okay, now it is okay. You can start.
Yeah.
Please. So some noise.
Yeah, somebody's mic is on and it seems that's
I will
okay.
check. I will check it. But I think all the mic.
Then you will be able to mute everyone and then
Mute all.
yeah.
Then that sound is again coming. Okay, madam, can you speak now?
Yeah, no, it's fine.
Okay, please start.
Yeah, I'm not able to switch on my video for some reason. I don't know what is wrong. Okay, anyway, so that is not the important thing now. Sujit, thank you for thank you for inviting me and giving a chance to participate in this. First, itself, like congrats to Sujit for organizing this kind of webinar series. And now this, I think it is the fourth one. So it's a wonderful job, like in this pandemic time. You're doing a great thing. And we are able to hear from the good scientists like Dr. Praveen. So thank you for giving a chance and for the great introduction. Of course, you don't have to give that kind of an introduction to about me. Like today's person is Dr. Praveen. So anyway, thank you for all those. So my job is basically to introduce Dr. Praveen. And yeah, though I don't know personally, I haven't we did not have any personal interaction earlier, but I know about you. I heard about you. I read some papers. So yeah, he's a very well-known scientist. from the probably Professor Ajay Ghosh's students. Now his senior has proved about the supramolecular chemistry, the importance of supramolecular chemistry by receiving the Shanti Swarup Bhatnagar Award of this year, Dr. Subhi George. So first itself, I wish Dr. Praveen, let you be the next person to receive this award. And so I come to my joke. Professor Dr. V.K. Praveen, he completed his MSc in Applied Chemistry from University of Calicut in 2001 and then did his PhD from CSIR-NIIST. I think at that time it was CSIR-RRL under the supervision of Professor Ajay Ghosh. He then joined in the University of Tokyo as a Global Center of Excellence postdoctoral fellow and then later as a JSPS fellow. Then again moved to Italy in 2011 as a Marie Curie fellow. So holding these two prestigious postdoctoral fellowships, he completed his third postdoctoral from Italy at ISOF CNR Belgrano. Then he returned to India and joined with Professor Ajay Ghosh again. And during that time, he received the Startup Research Grant for Young Scientists from DST, which allowed him to start his own research groups at the Photoscience and Photonics section at CSIR-NIIST. So in 2016, he got promoted as CSIR Scientist Fellow and a CSIR Assistant Professor. He became a Senior Scientist in 2018. His research interests include supramolecular systems chemistry with a focus on study on pathway complexity in supramolecular self-assembly, study of photophysical properties of fluorescent molecules, and development of these materials for security and sensing applications. Yeah, he published more than 50 articles in various journals, international journals of repute, very good journals, of course, and 4 book chapters. And his papers received over 6,000 citations and with an H index of 31 and I-10 index of 42. So this itself is showing the importance of his work. Published around 50 articles and in that itself 42 received an I-10 index. That means at least 42 papers were cited more than 10 times. So out of this 50 article, that means all papers, all the work he does is of that importance. So I'm happy to welcome you, Dr. Praveen, to this session. And as I said, all the very best wishes for the next Bhatnagar Award. We are looking forward to hear that good news. Dr. Praveen. Yeah, actually, I also was listening to Professor Len last week he was telling about from how the chemistry started. It started with the molecules and now it is with the supramolecular. I think you are also going to take us to that journey from Big Bang theory to the present systems. Everywhere we are using chemistry and the supramolecular chemistry. Yeah, Professor Praveen, over to you.
Okay, thank you, madam, for your kind words of introduction. And also, I would like to thank Dr. Sujit and Unnikrishan sir for giving me an opportunity to present in front of all of you. As madam has mentioned, as about Subhi Jacob George and Professor Ajay Ghosh, I was lucky enough to interact with all these people in the beginning of my research. Still, I am continuing my interaction with all these people now also. Because I also want to mention about Professor M.V. George, who actually started our department in CSIR RRL that time. Now it's a CSIR NIST. And he was giving us all the opportunity to do whatever we want in research. and he gave us a freedom to think. He gave us a freedom to be a little bit ambitious and achieve whatever you want in your life. And so happy to see that one in the initiative of some of the professors in Calicut. They are going to conduct some seminar series from Saturday onwards. I encourage all of you to attend. There, Professor Ajay Ghosh is giving a lecture. His experience he is going to share about supramolecular chemistry. And whatever Madam is told is correct, because after Dr. Ajay Ghosh, I feel Uday Maitra, Professor Uday Maitra and Dr. Ajay Ghosh now only supramolecular chemistry is getting another recognition through SUBI because of his very hardworking personality, as well as his very. I was so lucky that one I was one of the first junior, so he has inspired me a lot how to do research and what is his importance. Still we are continuing that relationship. Whenever I want, I can call him. He will explain me. Okay, so today I am going to share something about the recent development in supramolecular chemistry, which we call as a supramolecular systems chemistry. I will share the slides first. Okay. Okay, I think the slides are visible now. Please believe I will start. Okay. I think most of the students and teachers may be familiar with the terminology supramolecular chemistry, but we can see a new terminology we actually added in between, that is called as a supramolecular systems chemistry. And why, what is systems chemistry, why it is important, that you people can understand from through during this seminar. Supramolecular chemistry, as per the Professor Jean Marie Lagan's definition, it's a chemistry beyond the molecule. So we are going to talk about chemistry of molecules. That is what kind of chemistry molecules we are doing once it is synthesized, what kind of communication it is doing, and how this communication is leading to supramolecular organization or supramolecular self-assembly and hierarchical self-assembly. And as Professor Lan has mentioned, and this hierarchical organization has can lead to a very complex systems, and once it can lead to a thinking organism like us. So we are quite special in that perspective, because we know that when if you think human being, I will tell you in our perspective what is human being and what is its relationship with human being and supramolecular system chemistry or supramolecular chemistry. So all of you know that one chemistry is I put chemistry in between physics and biology. Biology is the study of living organisms and their functions and structures and a lot of other things. And if you do a little bit of biology deeper and deeper, if you try to understand the chemistry behind this process, then biology will slowly change its face into a new field. It will become a chemistry. So similarly, if you do in chemistry, if you try to understand more and more of the mechanistic details, and if you want to apply this material, this chemistry to make some material, it will just move into physics. Okay, that's why we place chemistry in between the physics and biology. And as all of you know that one now, there is no boundary between any of these science disciplines, because all of them are in the overlap. And in our courses, we can see physical chemistry is there, biochemistry is there. So I think chemistry is a field which is a connecting field, which can connect which can connect actually physics as well as biology. That is what I want to give an idea. So as all of you know that the birth of the molecules and materials all start from this lab. This is the way we create molecules, and this is the way we synthesize molecule. We can see a stirrer. We put a reaction. Since we are working on a fluorescent molecules or a molecule which has a strong absorption, we will start with a fluorescent molecule, and we will synthesize molecule. The next step is we have to purify it. So we will go for a conventional methods like extraction, and the left side you can see that column chromatography, which we normally use to purify the molecules, and we will make pure molecules and we will do their studies. Why we start with the pure molecule? Because then only we can compare their properties with their structure and property relationship. We can, otherwise we have no idea that why we got this particular property. And another topic which has a lot of interest in the last decade, that was polymers. Polymers actually are large molecules and macromolecule composed of many repeating subunits or identical or different. So due to the wide range of properties, synthetic and natural polymers play an essential role in our everyday life. You can see some of the examples. You can find polymers in adhesive, carpeting, rubbers, fabrics, paints, and etc. So I'm not going to talk anything about these polymers, particularly covalent polymers. I'm going to talk about another class of molecules, polymers. It's called as supramolecular polymers. So we will first say that one of the polymerization process, some of the polymerization process which are quite familiar, quite famous are condensation polymerization, where we'll use a dicarboxylic acid and a dialcohol. We make a polyester, and the radical polymerization will take a radical initiator and we will react active compounds. Then the radical reaction will propagate until it will propagate in a way that until it get terminated with another puncture or something. So these are the conventional method to prepare the polymers, different varieties of polymers. I took 2 examples to explain this one. So today we are going to talk about another class of polymers where the molecules are connected by supramolecular interaction. We call it as supramolecular polymers. So before going to the supramolecular polymers, I will just compare the properties of conventional polymers and supramolecular polymers. Conventional polymers has a less order because of the building blocks. Monomers are connected by covalent bonds and they have a high melting point and become viscous upon melting, and high temperature and pressure required for the processing of the viscous melt. And since these are formed by a covalent interaction, often their interactions are irreversible, and therefore self-healing and self-recovery properties are difficult to achieve. But coming into supramolecular polymers, the supramolecular polymers are made of small molecules, and then they have a high purity. Therefore, when it connected by through non-covalent interaction, they have a high degree of internal order. And the building blocks are connected through non-covalent linkages. Some of the very strong interactions like hydrogen bonding, electrostatic interaction, you can use it. And easy to form low viscous melt because of the high processability we can ensure. And they are easy to purify. And since the non-covalent interactions are reversible in nature, we can actually achieve self-healing behavior and elasticity as well as the strength. So I'll go to the next slide. 1 minute. These are some of the examples of supramolecular polymers prepared by different types of monomers. This is called as a quadrupole hydrogen bonding. That is two molecules are connected by 4 hydrogen bonding units, and it is linked with a small polymer units. But essentially, the linkage between 2 polymeric units were through hydrogen bonding, and this is one type of supramolecular polymer. Another one is called as the second one is called as a lipid amphiphiles. It's containing a polar head group as well as a lipid tail, and this self-assemble to form a nanofibers. And another one is a fluorophore connected with the quadrupole hydrogen bonding. Because of its containing chiral chain, this can self-assemble to form twisted nanoribbons, just like DNA helical structures or collagen triple helical structures. And the third one is the molecule is called as a hexabenzocoronin. It's actually called as a small piece of a graphene. It's called as a nanographene. It's a Gemini-shaped amphiphile. Is one part is the top part contains a glycol chain, and the bottom part contains a alkyl chain. So this amphiphile can self-assemble to form nanotube. This is one of the molecule. My professor was very famous on this molecule. Actually, his group. I got an opportunity to work on this kind of molecules. It's called as a hexabenzocoronin based amphiphile. This amphiphile can self-assemble to form nanotube, just like a carbon nanotube. But the difference is this nanotube, the surface outside and inside, we can introduce a different functionality. So this particular organic nanotube is very interesting to develop different kind of properties, just like photoconductivity or magnetic property as well as fluorescence properties. We can introduce different properties. Maybe it's quite difficult in the case of carbon nanotubes. And this is one of the most studied and one of the most studied example of supramolecular polymer from the group of Professor Bettmeyer. And this is a particular molecule called as a 2-urido-pyrimidinone. It can form a dimer through hydrogen bonding, and it is connected with a small linker. So this can form a supramolecular polymers, and the supramolecular polymers actually have a properties like a conventional polymers. I will show you this Professor Bettmeyer's group actually in collaboration with some of the companies. They actually develop some polymeric material. I just download one of the video from their website just to show you what is the application of this kind of supramolecular polymers. This is a company is called as a Supramolecular. That is a company they actually started. Okay, 1 minute, I will try to share. One minute. I think the video is visible to all. Okay.
Yes, visible.
Okay. This is one of the supramolecular polymers developed by this company. It's called as a Suprapolix. Actually, using this supramolecular polymer, they actually created a self-healing elastomer. And since it is a supramolecular polymer, it's a monomer units are connected by hydrogen bonding. We can actually ensure the self-healing property. That is what they are going to demonstrate here. We can cut it. Okay, this is a freshly cut surface, and you can connect it again. This is a kind of adhesion properties. So instantly build up original strength. So this is one of the advantages of this kind of a supramolecular polymers. Another thing they have developed is some kind of a surface. You can just make some marks. Just heat it, it will start to melt, and then it will cure. And the scratches will disappear. And another thing is there are some kind of a hydrogel they prepared, the same kind of material. So when you heat it, it will just start to melt, just like it will become like a water kind of things. Okay. I think my presentation is visible now. It's okay. Hello.
Yes, it is visible. But I think you can slideshow. Can keep it in slideshow.
Oh, okay. Because I'm not familiar with this platform, so I'm taking a little bit of time to understand it properly. Okay. Okay. In 1 So the mechanism of supramolecular polymerization we can divide into two types because of the how these molecules are assembled each other. Based on that one, we can call it as an isodesmic self-assembly and nucleation group self-assembly. And I will explain you what is isodesmic self-assembly. In the case of isodesmic self-assembly, the monomer units are connected with the equilibrium constant between the monomer is same, irrespective of the formation of dimer or trimer or it's containing a 10 units. So only one thing about the isodesmic self-assembly, you have to remember that one, the equilibrium constant for association constant for this one is same. But in the case of a nucleation growth model, that is a cooperative self-assembly process, it's just like a crystallization. Initially one equilibrium constant is there, that equilibrium constant is corresponding to a nucleation region. Then once it is a nucleus with a critical size is formed, then it will start to elongate. That is called as a red color. It is called as a elongation region. This called as a overall this process is called as a nucleation elongation process. And I will explain you what is its importance, how we can study these things in detail in the case of a supramolecular self-assembly of small molecules. The isodesmic self-assembly is called as a downhill process. There is no uphill process, that is some kind of a nucleation process is not required. But in the case of a cooperative supramolecular polymerization, the first process is an uphill process, energy is required. Then some kind of energy we have to give, then only that nucleus will form. Once it is a nucleus formed, then the next step is the elongation process in a downhill process. And this supramolecular polymerization process we can divide into two categories. In one category we call as a living category. It is called as a non-dissipative self-assembly because it's here what we will do is that one, when you create this self-assembly, it can lead to different kind of states. One is called as a kinetically trapped state, another one is a metastable state or thermodynamic state. So how we can distinguish this state? Just like in our organic chemistry, kinetic product and thermodynamic product, depending upon the energy we are giving, this molecule can self-assemble and can get entrapped. In the case of if it is energy barrier for conversion from one state to another state is very high, we can call it as a kinetically trapped state. If the energy barrier is less amount of energy, if you give, it can transfer from one state to another state. It is called as a metastable state. And another more interesting state is called as a transient assembly. In the transient assembly, something like that one too. Some molecules to assemble, we have to give some energy. So once we give energy, the molecule get activated, then it will start to assemble. Once we remove the energy, it will start to collapse again. So this is a special case of a self-assembly is called a transient assembly. They will exist for a very short time of time. If you see any of our biological system, we can find out n number of transient assembly, because whenever there is energy flow is there, this system will assemble and will exist. But once you remove the energy flow, then it will collapse. For example, you take human beings, as far as we breathe or as far as we give some energy input, for example, in our case, oxygen or water or any kind of input we give, we are existing. Once it stop, we will disintegrate, we will break apart, and we will disassemble to form to our elements, whatever carbon or other elements. So in one way, human being is one of the most complex transient assembly. Okay, so first I will discuss about supramolecular chemistry. This is one of the paper by Subi Jacob George, which when he was a postdoctoral fellow with Bettmeyer, he has demonstrated the pathway complexity. That is, whenever molecule self-assemble, how much complex it is. He demonstrated with the help of a small efflorescent molecule. It is called as oligophenylene, equipped with a hydrogen bonding unit. This molecule containing chiral chain. So what they observed that one, when this molecule self-assemble, we can see in the bottom part, it will form actually a disordered aggregate and form a nucleus. Then it will start to aggregate to form some helical structure. So most of the time it will form a left-handed helix cells. That is, in a spectra we can see that blue line corresponding to a left-handed helical structure. But in some cases, when the same sample, if you rapidly cool, then there is a change in the temperature, then it will start to form some disordered aggregate. It will start to assemble in opposite way. Sometimes it can lead to some right-handed helix also. How they monitor that one? They have done a concentration dependent studies. You can see there. we can see that at a higher concentration and initial time scale, CD reversal they actually notice. That is corresponding to the formation of a helix with a opposite hand ends. With the time, it is converting back to the left-handed helix. So how they demonstrated this one with the help of a achiral molecule. The first one is a chiral molecule. This they helped with the achiral molecule. What they did with the achiral molecule is that one they prepared achiral OPV. Then they used a tartaric acid derivatives as a chiral auxiliary, just like in our organic chemistry. So this chiral auxiliary can bind with the OPV hydrogen bonded unit. Then it will form a particular helical system. And then they removed the chiral auxiliary. Then they found that one this assembly is stable for a particular amount of time. Then with time it will convert to the opposite helix. That is what is there demonstrated in the bottom figure. Initially it has a right-handed helix, that is a line, and when they removed this one, it exists in a little bit of time. With the time it will convert to the opposite helix. This is the demonstrator. This demonstrating that one. Even one of the problem we are now facing that one. If you are doing any supramolecular chemistry, people are not giving attention to the supramolecular assembly procedures. They are simply, even if it is a solvent evaporation or a simply heat and cool, or if you crystallize it, just like a crystallization, if you are adding a one solvent to another good solvent to a bad solvent. These are all procedures, just like in organic chemistry. This procedure has some means. So you have to clearly note down the condition at which temperature you did this experiment, what was the room temperature, what was the concentration, and how much time you kept the sample for processing it. So each and every aspects are very important. So until recently, people were not giving attention to this kind of a methodology, because they simply say that when I got a left-handed helix, I got a right-handed helix, I got a fiber, I got a spear, but they have no clue that one why they got, and often they have a problem to reproduce this one. So it is very important that one, just like in a organic chemistry, we have to understand the proper self-assembling conditions. Once you optimize that condition, you have to note down, because of these conditions are required to create hierarchical assembly. Because if somebody wants to reproduce your result, somebody wants to make a more complex system, just like organic reactions, several organic reactions, people were taking and creating a very complex molecules. So if all the supramolecular chemists are publishing their work with a proper experimental procedure, anybody can reproduce that result, and then they can think about making a more complex system through non-covalent synthesis. So this is very important, and through this paper they have demonstrated, okay, you are making a left-handed helix, but you have a chance to get a right-handed helix if you are not doing things properly. I mean, if you are carefully not following the self-assembling condition. This is the pathway complexity present in this kind of systems. Okay, so then from our lab we have actually demonstrated, just like in a
Dr. Praveen, can I interfere?
Okay. Yes.
Your slide is not visible now.
Is not visible?
Not visible now.
Probably something changed that.
Okay. Now it's okay?
Yeah, now it is coming.
Oh, yes. Okay.
Yeah.
So this is one of the work 2017 we published. Here also we have noticed some of these complexities, because here in this OPV, oligophenylvenyl based systems, this can form a hydrogen bonding network and then self-assemble to one-dimensional fibers. So what we notice in this case also, depending upon the condition, we can end up with a different kind of helicity. So what may be the reason? So we just investigated in details. That is what I am going to discuss now. And what at very low concentration, 5 into 10 raised to minus 6 molar, these molecules are self-assembled to form a left-handed helix, and at a 10 times higher concentration, the same molecule self-assembled to give a right-handed helix. Initially, it was very tricky, because we are not sure what is happening. So always we think that is a mistake we made. So we repeated this experiment. We thought that we can reproduce this result. Low concentration, we are getting one kind of a circular dichroism signal, and higher concentration, the CD signal is reversing. So in order to check that one, we made a molecule with the opposite chiral handle, that with R. And there also we could reproduce this. So it is clearly showing that one. So something is there. The system wants to convey some of this message to us. We actually need to identify what it is, then we have to follow it. This is what is meant by systems chemistry, because you have to understand the chemistry of that particular systems in detail, then we can explore its property. So what we did a detailed temperature dependent CD cooling curves. For this purpose, what we do, we made different concentration, 5 into 10 raised to minus 6 molar to 5 into 10 raised to minus 5 molar concentration, and we monitor the CD changes at particular temperature. At the same time, we kept the cooling rate at 1 Kelvin minute, because this is very important. If you change the cooling rate, sometimes the properties can change depending upon the condition. Then the kinetics play a role. So if you want to do all the process under thermodynamic control, you have to reduce the cooling rate as low as possible. In this particular case, 1 Kelvin per minute is okay. Then we cool the sample and simultaneously we monitor the cooling curves. And we found that one in the case of a 5 into 10 raised to minus, it's completely a negative CD signal. That means that it's a left-handed helix. But as we increase the concentration, we can see a dip. That dip is corresponding to negative. Then with temperature, it is changing to positive. And as we increase the higher concentration, dip become very small, because in the case of 5 into 10, the very small dip we can see, because at higher temperature itself we change it to the right hand. So that means a kinetic process is involved in here, because with the temperature, with the concentration, one particular helical structure is formed. In this case, a left-handed helix is formed, then it's slowly converting into a right-handed helix. What may be the reason? In order to check that one, we monitored the morphology of this system. At very low concentration, we found that the left-handed helix, you can see that M helix, and at a moderate concentration, we found that presence of both left-handed and right-handed helix. In this particular case, we identified that one, the right-handed helix are formed by the supercoiling, just like our proteins or collagen triple helix. Once a helical structure is formed, that is showing a M helix, when it is started to form a coil, so then it will start to coil in opposite way, and it will end up with the opposite helicity. So this is the one particular aspect is completely ignored in the case of a supramolecular chirality, because people have not considered the importance of supercoiling in the observed CDC, observed circular dichroism properties of this particular molecule. Through this work, we actually demonstrated the importance of supercoiling in getting the CD signal. And at a higher concentration, we got actually a bundled supercoil structure, which has a right-handed helix. So we started with the M helix, and when it started to form hierarchical assembly, it's started to coil in opposite way, and it end up with the opposite helix. So this is same like in our biological system, any proteins or DNA or collagen triple helix. In a lot of cases, biological system, this supercoiling is playing a big role. And then we did a lot of control experiment to prove this one. One experiment we did, we took a P super helix, that is the supercoiled aggregate, we took it, and we started to do a depolymerization. In this case of a supramolecular polymerization, to do the depolymerization, we have to add a good solvent. In order to maintain the total concentration same, what we did is that one, we dissolved monomer into good solvent, and we added in a different fraction. We found that at as we increase the chloroform fraction, the P superhelix is started to decoil, and slowly CD signal is changed. At 15% of chloroform, it is changed to completely M helix. And again, when we increase the concentration of chloroforms, then this M helix started to disassemble back to the monomers. So there we can see that at CD signal is completely vanished. And another experiment we did a dilution experiment. We took a P helix and we diluted with decane, and we maintained the total concentration is changed, and we found that one CD signal has no change because of there is no decane has no role. Decane cannot decoil this kind of a system. And then we did a very tricky experiment. We actually created two kinds of a helical structure from the same molecule. First, we created a hemihelical structure with a left-handed helix at very low concentration. For example, the first molecule with S at very low concentration, it forms left-handed helix, 5 into 10 raised to minus 6. But the same molecule, when at higher concentration, form a P superhelix, and what we did that this P superhelix is prepared at 5 into 10 raised to minus 5 molar. We diluted and we created a same concentration, 5 into 10 raised to minus 6, but its helicity is opposite. So then we got an opportunity to compare both the properties at same concentration but different helix. In the case of P superhelix, which is prepared by the dilution, we found that when we increase the temperature, it started to decoil, and decoil it to form M helix. Then finally it is a monomer at higher temperature. But in the case of a M helix, since there is no supercoiling, it is directly disassembled to monomers, and we can see that at some stages at 330 Kelvin we can see that the hem helix depolymerization is matching. This is one of the best experiment we did, and in summary, depending upon the concentration and condition, the coiled molecules can show one kind of helicity. When it is supercoiled, it can give rise to different kind of helicity, and the whole process we can explain. First it will form a monomers and it will nucleus and it form a small helical fibers. Then it will start to coil and form a P superhelix. And in our case, we were lucky enough that one initially formed the helical assembly and finally formed the helical assembly as different CD signals. So we could follow this pathway complexity in this supramolecular self-assembly process. So the most satisfying things whenever we publish any paper, when people are started to quote our work, and they also try to explore the similar kind of concept in their system. This is one of the example. After our publication, this paper is already got nearly 60 citation, and a lot of scientists also started to cite our paper. This is one of the work. In this work, they also notice, for example, you can see that bottom figure. The monomers are first from a metastable monomers, and then it start to aggregate, and this aggregate, depending upon the condition, is from a superhelical aggregates. Whatever we observed in our system, they also start to observe in there. They also try to explain similar way. We actually try to unravel a particular property in the case of a supramolecular self-assembly of a chiral molecules and its hierarchical assembly. So another interesting example is from my professor in Japan, Professor Takuzo Eda. It's a very interesting example. This is first time they have demonstrated living supramolecular polymerization in the case of a supramolecular polymers. First of all, all of us know that this is a fullerene. If you cut from as shown here, we can end up with a molecule is called as a uranulene. This molecule has a tendency to flip each other. Ball-to-ball inversion is possible. In order to prevent this ball-to-ball inversion, they actually introduce amide group. There are 6 positions are there shown in yellow color. They actually introduce 6 amide groups. So this will actually form an intramolecular hydrogen bonding, and then it will prevent the ball-to-ball inversion. So these are all called as a conformationally dormant monomers, because it cannot self-assemble because of the hydrogen bonding units are formed through intramolecular hydrogen bonding. So actually it will prevent the ball-to-ball inversion. At the same time, the monomers are dormant. They cannot self-assemble. Okay, this is the way the intramolecular hydrogen bonding is formed. In the way the molecules are locked, it's called as a dormant monomer. And then what they created as some initiator from the same molecule. For that purpose, what they did, in order to prevent the intramolecular hydrogen bonding, they actually methylated that amide bond. That is, we can call the molecule first molecule I, and Me, we can see that one. This molecule they used as an initiator. What they did, okay, so then initiator they were added to the dormant molecule. The dormant molecules forming an intramolecular hydrogen bonding, so they are not in the state to assemble. When they add initiator, what will happen? We can see here. Okay. The initiator molecule will attack to the dormant molecule. Then the hydrogen bonding it will break. So the dormant monomer is active. Then it will start to self-assemble. So then it will form a assembly. Just like in a chain growth polymerization, here monomer is made as an initiator by methylating the amide bond. Then that is used to add as an initiator into another system, which is a dormant instead, because it is forming intramolecular hydrogen bonding. When this molecule attack from hydrogen bonding with the dormant monomers, actually it is actually activating the monomer. Then it will start to form intermolecular hydrogen bonding and supramolecular polymer. So they first time they actually demonstrated the concept of living supramolecular polymerization. So coming into the interesting work, so I may take a little bit of time to explain this one. This is one of the experiments I was talking about, nucleation growth model. So is it possible to visualize these things? For example, it may be very interesting for students to know that how this I was talking about kinetically trapped state. I was talking about metastable state. I was talking about thermodynamics. Is it possible to visualize also? This work, Professor D. Kola and co-workers has nicely demonstrated that one. This metastable state and kinetically trapped state and thermodynamics we can actually visualize. This particular platinum complex, depending upon the platinum-platinum interaction, it can actually form 3 different kind of assemblies. Depending upon the emission, it can form a if the platinum-platinum interactions are very high, it will form a red emitting particles. That is a top one. If the platinum interactions are moderate, it will form a kinetically trapped metastable aggregates. Why it is called as a kinetically trapped metastable aggregate? Because of this green color aggregate, with time it converted into a blue color aggregate where the platinum-platinum interactions are very less. So this is schematically this show. Depending upon the emission, actually these molecules are divided into 3 categories. First one is a red emitting kinetically trapped metastable aggregates, then the green emitting metastable aggregates, and the blue emitting thermodynamic aggregates. So what they demonstrated, depending upon the condition, I actually need to share some of the videos. 1 minute.
Dr. Praveen, your mic is mute.
Is it okay? Is it audible? Hello?
Yes, yes.
Okay, audible.
Okay. I think my video is
gone. When the slides
I will share it. I will share.
You can, there is a start video option. You can.
Settings.
No, start video. It is just to show like only we can
Yes.
the slide. The slide itself we may have to.
Okay, now. Okay.
Yeah, now it's coming.
Is it okay now?
Yeah.
So this is one of the nice experiment they did. Actually, they have created a condition where they can clearly see that the existence of red emitting aggregate and the green emitting metastable aggregate, and with time how it is converting into a thermodynamically stable aggregate. So they actually monitored this whole process through the help of confocal fluorescence microscopy. So at a particular condition, these molecules actually form red emitting aggregates. With time, this red emitting aggregates is converted to a green emitting metastable aggregate. We can see that one simultaneously blue emitting thermodynamic aggregates were also forming. Okay. As the time progresses, we can see that one the number of red emitting aggregates are less because it's slowly converted into a green emitting aggregate. Then this green emitting aggregate is slowly converting into a blue emitting aggregate. We can see that one. In some of the cases, we can see that one. So this is in a particular condition. When they change the condition, then they change the condition to another condition. Example. When they change the condition to another, here it is interesting thing is that one, here they can actually convert the thermodynamic stable aggregates, that is a blue emitting aggregate to a green emitting aggregate. What they did is that one, they excited the blue emitting aggregate with a 405 nanometer laser light. Because of the laser light will excite all the molecule, it will go to the excited state, that will produce some thermal energy. Because of the thermal energy, they start to reassemble and then slowly converted it to its metastable form, that is a green emitting aggregate. For that purpose, they require a very high source of laser light, and they irradiate with the blue aggregate with the laser light, and we can see what is happening here. Because with the time it is getting converted into green emitting aggregates. So these experiments actually clearly demonstrating that one, just like in organic chemistry, with the condition, with the concentration, with the temperature, we can also achieve different kind of a product, like a kinetic product or thermodynamic product, etc. So finally, I will discuss about the transient assemblies. As I mentioned here, in the case of a transient assembly, what actually required, we have to give a fuel. So initially, one inactive monomer will be there. So when you give a fuel, then it will form an active monomer. Then the active monomer start to assemble to form a transient state. Once you remove the energy or fuel, or if you stop the input of fuel, then it will start to disassemble again and to monomer. So the existence of these assemblies in a transient nature, that is called as a transient assembly. It's a very nice example, very simple work. They demonstrated dicarboxylic acid because of the charged anion, it cannot self-assemble. They methylate this compound, because once you methylate, because this molecule will be hydrophobic in nature, and the condition is water, so it will start to assemble. So once and what they did is that one, they adjusted in a condition in such a way that one, the hydrolysis will take place simultaneously. So they added some hydroxide anion. So once it is formed a methylated derivative, it will exist for a transient, a short duration of time, but at the same time it will hydrolyze back to the monomer again. So this is a cycle. This is a transient cycle. This is a very simple experiment, but they actually demonstrated the existence of a transient assembly. So one of the elegant examples from is from the research group of Professor Grabowski and Raphael Klein. Actually, they demonstrated the transient inks with the help of this transient assembly. For this purpose, they have taken a gold nanoparticle and functionalized with azobenzene. As all of you know that one, azobenzene is a photochromic molecule. If you irradiate it with UV light, it will transfer into cis form. When compared to the trans form, the cis form is polar in nature, so that is why it will start to aggregate. Okay, or that will eventually lead to a change in the optical properties of gold nanoparticle. So when the molecules, the gold nanoparticle is functionalized with a trans-azobenzene, they will be more like a dispersed form. When you irradiate the UV light, it will convert it into cis form. Then it will start to assemble. Then eventually it will lead to a change in the gold nanoparticle absorption properties. That is where demonstrated. So they took a solution and they irradiated with the UV light. Within time, it is converted into cis form. We can see that when a change in the absorption corresponding to the gold nanoparticle, we can see a long wavelength feature is started to appear. So by using a photo mask, actually they actually demonstrated how we can utilize this particular transient assembly to create some transient images. So actually they dispersed the gold nanoparticle in a PMMA gel with the help of a mask. They first UV irradiated. Wherever the UV light falls, there it will convert it into a cis form, and it is from the gold nanoparticle start to aggregate. Then we can actually create an image. So this image, they actually created an image of a tiger, or in the bottom part we can see that one is just like our Mission Impossible movie. It's a transient message. Once you read it, then it start to perish. So we can see that this message will self-erase in 1 minute. We can see that within 1 minute of time, it is completely, because of this back isomerization is happening. Whatever azobensin is converted into cis form, with time, under normal condition, it can convert back to the trans form. So that will convert the whole system into the trans system. Then gold nanoparticle lose its properties. So in the initial case, first they irradiate with the UV light. The trans azobensin is get converted into a cis form that will trigger assembly and simultaneous changes in the absorption properties of gold nanoparticles. So we can see an image with the time. The cis is getting converted into a trans state that will again disperse the gold nanoparticle. Then it will lose its characteristic absorption features. Then we can see that erasal of image, and n number of times we can repeat. That's by using UV and visible light. In the case of visible light, the cis azobensin will convert to transform very fast. So with the UV mask, they created azobensin image, then visible light, it erased. Then again with the UV mask, they created another image, and with the temperature and the UV mask, they actually created another image. And then we can repeat this n number of times. It's a clear indication, very nice demonstration of a transient assembly, how we can utilize to create some kind of a transient images or transient ink. Coming into this kind of a transient assembly, we actually also demonstrated Dr. Ajay Ghosh group, and I was working with Dr. Ajay Ghosh. This work they have actually demonstrated is called as here we actually created a transient images with the help of a water. For this purpose, we have taken 2 molecules. It's called as OPE molecules. One molecule is connected with a amide linkage, and another molecule is connected with the ester linkage. What is the characteristic feature of this molecule? This molecule will start to aggregate in THF water mixture, and THF it is showing a monomer. And depending upon this condition, this molecule can show different emission properties. In the monomer state, these molecules are blue emitting and it's very less emissive, and compared to the aggregated state. Once it is aggregated, its emission is shifting to the red region. We can see a cyan emission. The both molecules are showing the same property. But in the case of a first molecule, the molecule with amide linkage, what we have demonstrated that one, if you drop cast these molecules on a filter paper and then put a water, we can see that one, the color change is there because of this aggregation changes. And the presence of water, these molecules interacting with the water, and then it will create a different state which is emitting in the cyan state. When you dry the air, you will go back to the blue emitting state. And by using this water as an ink, we can actually create some images, and these images also, as far as water is there, the image is visible. Once you remove the water, it will go back to the original state. And here also we can, the self-erasable image we have written with the help of water as an ink, and with time it's slowly started to erase, and within 4 hours it is completely erased. So we can actually repeat these experiments a number of times. And we can use this ink as a security material. If you paint this ink with any documents or a currency note, and you know that one in our currency, several kind of security features. That fluorescence is one of the feature. But the problem is that the current fluorescent feature, anybody can reproduce the similar kind of fluorescence, because only one condition is that it has to fluorescent when you illuminate with a 365 nanometer. But this kind of a special type of fluorescent material which can give rise to a different emission in the presence of some stimuli like water. Water is a very common stimuli, common stimulus we can easily utilize. So when you touch with the water, you can see that one, the fluorescent material can show different color. So this kind of material may be a future security kind of a material we can develop. Such studies are progress in our lab, because for a commercialization we have to identify very small molecules which can retain the same properties, and how best we can retain these properties for a kind of making any security application. Because you know that one, counterfeiting is one of the most dangerous kind of a crime, because you know that one, the large number of products are counterfeited, and we have no idea that whether we are buying it from Amazon, or in case of Amazon or some other companies, whether we are getting the original one or not. So we actually require new security feature to make sure that on this security the material which we received is original or not. And this is the mechanism. Initially, this molecule is forming a hydrogen bond network that is in a blue in color. So in the presence of water, the water can go and interact with the hydrogen bonding unit. Then it can break the hydrogen bonding. That will allow the molecule to slip each other. Then it will form a J-type aggregate. It's called a slip aggregate. Finally, it will end up with a cyan emission. So another technique we did, we actually took two different molecules. PE1, as we know that one, it can show initially blue emission. Then with the time, when you add water, it can change the color to cyan, and with the time it can come back to the original fluorescent. The second molecule with the ester linkage, it will show only cyan emission. And the third molecule with the alkyl chain, it will show only blue emission. So what we thought about it, whether we can create any security labels, for example, a barcode. It's called a two type of barcodes we get. So we will take the first, the first molecule is first and third molecule. So when we print it, both molecules initially show blue emission. So this barcode is not readable. But if the moment you touch water, the first molecule will change color to cyan. Then the barcode is more become visible. It's called as a forward barcode. In the case of a first and second molecule, initially because of the color contrast, the barcode is readable. The moment you add water, the first molecule will change the color to like a second molecule. Then the barcode is not visible. This is called as a reverse barcode. So I will show you some of the videos. I think the video is visible. Okay.
Yes, it is.
Yes.
Yeah. This is a mask logo. Yeah. The logo is under daylight condition is not clear. In the mask logo under UV light. But if you spray with water, and actually the logo will become visible. The next the fluorescent paper under daylight. Fluorescent paper under UV light, the blue in color, and using water as an ink, actually you can write. And by using a daylight, it is not visible. and erasing the letters using a hot air. So if you heat it as the water evaporates, this is actually a transient assembly. The letters are gone. Okay. And the mechanism for the self-assembly we can explain. Initially it form a blue aggregates, and the water molecule is entering into that one because of the glycol chain. It can interact with the hydrogen bonding, and it will break that hydrogen bonding. That will allow the molecule to slip each other, and the slipping will lead to a fluorescence stage. So this state is the whatever state it is formed in the presence of water is actually a transient assembly, and this transient assembly, once water is removed, it can go back to the original state and it can retain its fluorescent property. And then another one is the barcode. Actually the barcode we created, you can see that one. As the water level rises, the barcode is become readable, and we can actually read it with the help of a mobile. Then we can actually, the message is written as Satyameva Jayate. I will play once again. The barcode is initially not readable, and the water rises, the barcode become easy to read, and the barcode is detected and it is actually decoded the message. Okay. I'll go back to my presentation. So I almost finished my presentation. I just want to share some of the latest development in our research group. This is some of the novel dyes we have actually developed. So I'm not disclosing its proper structures, because this molecule can show different kind of emission from blue to red. And what we found that one, this molecule is also emissive in solid state. And another interesting thing about these molecules are, so if you grind it, you can see that fluorescence change. Initially it is a green emitting. If you grind it, the emission is changed to orange emission. This kind of fluorescence properties, this is a transient fluorescence properties, are called as a mechanochromism. But in our case, very interesting, if you stop grinding, it will go back to again to the original state. So we can actually repeat this one. So this is called as a isothermal mechanochromism, because we don't need to apply any kind of external stimuli like a heat or a solvent. When you are applying pressure, the fluorescence is changing. But when you stop it, it will go back to its original state. And finally, some of the interesting assemblies, sensors we have developed is a recent sensor. We developed a carbon dioxide sensor, and this particular molecule is aggregation induced enhanced emission active molecule. That means when the molecule is the monomer state is not emissive, when it is aggregated, it will start to emit. And this we pick this molecule with a particular purpose, and it is attached with a particular moiety that is a tertiary amine moiety that can interact with the carbon dioxide. So once in the presence of water, what will happen? The carbon dioxide will form a carbonic acid, and the carbonic acid will protonate this amine. It will form a NH plus, and the bicarbonate will be anion. So since from a neutral molecule, when it converts to a charged molecule, it will disassemble back. So what we can see that when initially, because of the aggregation, this molecule form a orange emission, but we purge with the carbon dioxide, presence of carbon dioxide, it will disassemble back to a monomer state, and then emission will quench. But if you purge again with the nitrogen, the carbon dioxide will go, and it will again come back to the original state. That is in that perspective, it is some kind of a transient assembly, but we can utilize for a sensing purpose. Okay.
Thank you, Dr. Praveen, for taking us to the fascinating world of supramolecular chemistry. It's really wonderful to see your exciting research. Actually, it is giving us a strong message that like innovative thinking always leads to inspirational research, actually. It's very, really fascinating. And you have said it was really good. Like you started with the basics and then went on to say about all the self-assembly, transient assembly, all those came to interesting applications like how these kinds of systems, supramolecular systems, can be used for the security level and then also the sensors. It's really a wonderful talk. So now we have actually a few questions. The first, I have a question like I want to ask is, so you said about the helical structures, which with the temperature it is changing, with the concentration it is changing, R to M helixes. So with the temperature, you could change the helical structures, right?
Yeah.
So is it a reversible?
1 minute, I am talking. My computer has some problem. Now I am talking into my student's account, Agil. Okay.
Okay.
So can you repeat the question again, because in between my computer become shut down.
Oh, okay, fine. You were saying about the helical structures, the M helix and R helix. With the temperature, you are changing these helical structures. Temperature as well as the concentration. So with the temperature, with the concentration, you said it is reversible actually. What about with the temperature? Is it a reversible change?
Yeah, of course, it is a reversible change, because with the temperature, we actually demonstrated, because if you change with a P superhelix, that is, at a higher concentration, we can get a P superhelix. You increase the temperature, it is slowly converted into a M helix. Then finally, it will convert into a monomer state. So both by using concentration as well as temperature, and also by depolymerization by chloroform, we can actually convert.
Yeah, but I think you were saying like upon increasing the temperature, it changes to R to M helix and then to monomer. So my question is like, if you are cooling back, is it going the reverse way, like monomer to M helix? Is it?
Of course. It will go.
Oh, okay. So repeated heating cooling cyclic. So if you are giving, you can reverse. So it's a completely reversible assembly.
Yes. That is why supramolecular polymers advantage is there. It is not like a covalent polymers. You can actually break and you can reform. In the case of it is a chiral supramolecular polymers. So we actually started with a higher concentration P supramolecular helix, and then either use temperature or a concentration or some kind of a depolymerization by using a polar solvent. We can actually decoil, and first it will form a M helix, and then it will depolymerize to monomer. Then if you again cool it, it will go to the same pathway, because it will first form a M helix, then it started to coil. So the whole process is reversible.
Okay. Yeah, one more question I have is like CO2 sensors you are saying, is it selective? Like selective versus...
Because this is selective to carbon dioxide, because it is a kind of a reaction based. So whenever carbon dioxide is there, the carbon dioxide is interact with the water. I mean, as all of you know, that when it can form a carbonic acid, and then the acid will protonate that particular nitrogen. Because of the reason the emission changes is there. So in that perspective, it senses it is selective to carbon dioxide, but can interfere with a carbon disulfide.
Yeah, that's like that. Interference. Interference will be there. Even sulfur dioxide.
Other solvent, other gases like nitrogen or argon, it has no role. But carbon disulfide can interfere.
Sulfur dioxide or NO2 kind of.
NO2, I don't know, but this carbon disulfide is known to interfere with this.
Okay. Yeah, actually, we have a few questions from the participants. Ashwati, you can unmute and ask your question. Sujit, they can unmute, right?
They can unmute.
I think they can unmute.
They can. Yeah. Maybe you can ask her.
Yes, ma'am. Hello?
Yeah.
Yeah, you can ask the question. Yeah.
Okay, ma'am. Okay. So my question is that you have talked about supramolecular polymer-based fiber in the beginning, right?
Yeah.
So I would like to know whether it has a better mechanical property compared to that of the natural fibers or the fibers which are now available in the market, like rayon fibers and all.
In the case of a quadrupole hydrogen bonded, because if there is a 4 hydrogen bonding units are there, that is equivalent to 1 covalent bond. If that particular polymer has a properties very similar to some conventional polymers, that is what is I demonstrated in the first. So that is a special case. Now, if the weak interactions are very less, for example, if you have only one hydrogen bonding, we cannot compare its properties with a covalent polymer because there's no match. But if you have 4 hydrogen bonding units are there, then it will be somewhat similar to 1 covalent bond. Then it can match the properties of conventional polymers. So what I want to tell you that one, it's quite difficult to replace the conventional covalent polymers because it has its own advantage, its own drawbacks. But there are some places where we can use supramolecular polymers. For example, I was listening one of the lecture by Reliance Industry. So if you go to Reliance Industry, their advice board, Professor Jean-Marie Lian is there. So one of the questions, one of the problem they face whenever they make any tires, the tire at higher temperature, there will be some crack will be there. So how you will solve that issue? Because if the conventional polymers has no self-healing properties. So what this advice board, they actually requested them to use some kind of a supramolecular polymers along with the conventional polymers. So when you cool down, if it is a supramolecular polymer, it can actually heal that issue. So wherever we can use supramolecular polymers, some kind of a reversibility if you want, or self-healing properties, or if you require adaptivity, or if you have soft material, so then there you use supramolecular polymers. And if you want to really replace conventional polymers with the supramolecular polymers, then we have to do a lot of synthetic procedure. For example, if you want to introduce a quadrupole hydrogen bonding, you have to put enormous amount of synthetic effort, and that monomer maybe not commercially viable. Because if you go to industry, they actually want cheap material because they don't want to invest on a material which has a very high cost for their product. So actually, we want a cheap product. So there will be a trade-off is required because of covalent polymers and supramolecular polymers. But supramolecular polymers are especially advantageous if you want to use it in biological systems. They are more adaptable. They are more, that they are less toxic. So we can use it. Or some other purpose we can use it, supramolecular polymers. It is not required. It's my feeling it's not required to replace or compare covalent polymers with supramolecular polymers. These are two different entities. They have their own advantages and disadvantages. This is what is my feeling.
Okay, thank you, sir.
We have next question is from Dr. Baby. Baby, maybe you can unmute and ask your question.
Hi, sir. So this is me, Baby. Thank you for the excellent presentation, and also it's a very good, excellent work. And so my question is, what could be the driving force or the reason for the supercoiling is happening in the opposite direction, opposite chirality? So is there anything to do with the amide unit, or you have in the latest slide you have shown that the amide group has that?
At present, this is one of the interesting questions. I really want to answer it in my future work. So I have some plans to make some molecules where I can demonstrate it. So what clue we got it? It's called as a secondary nucleation. Just like in a biological system, once if you form a M helix, or it not necessary to be M helix, initially one helical structure is formed, then it contains a lot of alkyl chains are projecting outside. So that can actually trigger the lateral interaction. So then it will start to some coiling. So what we feel that the supercoiling is mainly driven by the secondary nucleation triggered by the lateral interaction between the alkyl chain, not amide hydrogen bonding, because amide hydrogen is buried inside the helical structures. So then only alkyl chains are projecting outside. So this alkyl chain can interact each other, then it can lead to some supercoiling. So this is reported in literature, but very rarely demonstrated. Actually, we are designing some system to clearly demonstrate the role of alkyl chain in supercoiling.
Is there any evidence for that? for that alkyl coiling is itself is acting for the secondary chirality?
It is not much evidence because other than alkyl chain interaction, what else can interact each other? Because of these helical structures formed by the pi-pi stacking hydrogen bonding interactions. So the pi-pi stacking as a part of a pi unit is buried inside the helical structures, and then what is projecting outside is only the alkyl chain. So
Yeah, but
two helical structure, if they want to communicate each other, there is only one way, is that lateral interaction, that is through because the thermodynamic parameters are giving a clue that whatever values we are getting at a higher concentration when it is from a supercoil, so it clearly give an idea that lateral interactions are driven by some other things, most likely hydrophobic interaction driven by the alkyl chain interactions. So at this moment, this is only an assumption, and I have to verify this one again with designing another system. Because I think it's a good clue for us to do the future studies. So definitely we will look into that one.
okay, thank you.
Dr. Minimol,
Yes, ma'am.
you have a question.
Sir, it was a very good presentation. So I have a question. Towards the end of the slides, you presented the fluorescence change when you apply pressure. So what is the exact mechanism behind
Because
this?
if you look at the literature, you can see that these kind of systems are called as the mechanochromic systems. Normally people explain it is a transition from the crystalline to amorphous state. Because initially the system may be crystalline. So when you are applying, the packing will change. That will different chromophore packing. The system will become amorphous. So when you keep it, it will come back to again the crystalline. So this is the one mechanism people are normally discuss. So if you want to convert a crystalline state to a mechanical amorphous state, you can apply pressure. So if you want to convert back the amorphous state to crystalline, you have to apply temperature. So you have to heat it again, then you melt. Then only you will get back the original state. Otherwise, you have to add solvent, so it will crystallize back. But in our case, I feel the mechanism is different. So the molecule is containing some rotatable group. So whenever you are applying the pressure, so this molecule, that group become more planar. That will lead to a more conjugation and leading to a redshift. The moment we release the pressure, so it will again get a freedom to rotate again. So it will come back to the original state. So that is why we are getting a isothermal mechanochromism. We don't need to give any kind of stimuli. We don't need to apply. Ordinary condition, it will come back to the original state. So we cannot use the conventional mechanism which normally people use, and very rarely this kind of isothermal mechanism, mechanochromism is visible, and our system is one of them. And we are now doing theoretical studies, as well as we have prepared n number of derivatives to prove our theory. So the most probable mechanism I told, it is because of the rotation. When you are applying pressure, that particular moiety become planar. That will part of the conjugation length, and it will lead to a redshift in emission. The moment you release the pressure, it will again get the freedom to rotate again. It will lead to a disruption in the conjugation. It will come back to the original state. That is what we are feeling.
Okay, that means when you rotate the molecule, you are saying there will be some band gap.
Some particular part of that molecule. It is just like a molecular. In our case, a particular phenyl group is there. That is not in a conjugation with the chromophore unit. The moment you apply pressure, that will become planar. Then it will part of the conjugation. Then it will extend the conjugation. It will lead to a redshift in emission. So we are always seeing a redshift in emission. So that is one of the clue. And the crystallographic studies also showing that particular group is not in conjugation with the remaining chromophores. So, but when we are applying pressure, so that energetics we are now studying through theoretical calculation, and also by designing several molecules without phenyl group, with some kind of a breaker. That is for that purpose we require additional studies. We really need to develop new molecules that we are doing currently. That work is not complete yet.
Any other questions from participants?
Dr. Sandhyarani, I have a question.
Yes, sir. Yes, sir. Nikishan, sir.
Thank you. Dr. Praveen, can you hear me, Dr. Praveen?
Yes, sir. Yes, sir. Yes, sir.
A wonderful presentation. Great. It was very pleasant to listen to you on supramolecular systems chemistry. Particularly, we love to listen to you on helical supramolecular architecture and fluorescence modulation. I have one question. Actually, two questions are there, but the questions are interrelated. When you said of helical supramolecular architecture modulation, the concentration dependency and temperature dependency. Typically, high temperature gives you a right-handed helix and a low temperature gives you left-handed helix. Did you observe any threshold value for concentration? From which concentration to which concentration the left one will be dominating?
No, we actually did the first experiment we did when we found that at a 5 into 10 raised to minus 6 moles concentration, we found a left-handed helix. For a particular molecule. When we increase the concentration to 10 times, 5 into 10 raised to minus 5 molar,
that is the threshold.
An opposite.
That is the threshold.
No, that is the opposite one. But in between, if you take it in between concentration, we found that one both is existing. So we actually did concentration dependent studies to understand whatever you told the threshold, because it is very important, because we have to set two extreme conditions, also intermediate conditions.
Okay. So in between you got something which contains both the system.
Both sides. So, morphologically, we found that the existence of both M and P helix at the intermediate concentration. For example, 5 into 10 raised to or 1 into 10 raised to minus 6 or something like that. The in-between concentration.
Right. And another question related to that is, in supramolecular assembly, do you find a threshold value of molecular mass? Because the monomer is having a specific molecular mass upon associations. When the associations begins, gradually molecular weights will start getting built up. So
that depends upon that the molecular mass of the self-assembly for our system is maybe difficult to determine, because actually we don't have any particular setup to determine, and also at the same time the system has to be stable enough to study that one. In fact, depending upon other systems, they can calculate this the molecular weight of that supramolecular polymer, just like in any conventional polymers. So that kind of studies people are doing, but that requires a different set of molecules.
Conventional GPC may not support us.
Yeah, conventional GPC may not work in our case, because one problem is that almost all our molecules are solubility is a problem. When we are injecting this molecule, the GPC, that will contaminate the GPC and fluorescent molecules. So we particularly utilize fluorescence absorption to probe this molecule. But my professor in Japan, Takuso Eda, he actually is a hardcore polymer chemist. So they are depending upon the system, whatever sir is asking, they were studying its molecular weight, polydispersity, everything they are studying, because their systems are more facile to withstand all these conditions, and they can actually calculate all the parameters.
Thank you. One more question. If Dr. Sandhyarani permits, I will have one more question.
Yes,
Dr. Praveen, this is the question. Could you similar to the bigger systems, the glass transitions are very important. Typically, glass transitions, melting, etc. So for the supramolecular assembly, did you observe any? Did you do any DSC scanning or something like that to evaluate phase transitions?
DSC studies, DSC is one method to study the supramolecular polymerization, but the thing is that we required 5 milligram, because it is not like we are not making this molecule in a large quantity, but whatever it is required. For example, if you want to make a block polymers, this is one of the method to study, because we can clearly see that the melting point will be different depending upon what kind of block it is formed, because that is another trend in supramolecular chemistry. Just like in a conventional polymer chemistry, we can actually create a block supramolecular polymers. Then if it is formed a co-assembly through block polymerization, and then there will be definitely a change in the melting point will be there. Then corresponding thermodynamic parameters will be different. So every time we have to isolate the supramolecular polymers, and then we have to dry it up, and then we have to conduct a DSC study. But a DSC, of course, it is possible to do that.
Thank you very
This
much.
is a
Thank
very
you
good
very much.
technique.
Yeah. Thank you, Dr. Praveen. Thank you very much.
Thank you, sir.
Yeah, okay, now it's very late. Like, you spent almost 1.5 hours, more than that, with us. It was really wonderful to hear. And lots of messages are coming, like excellent presentations. So I hope there are actually a few more questions. Hope you will be available to them if they are interacting with you personally. Some mail ID or something, if you can give, that would be good. Like for our students also, like it will be really good if you could share your contact details. I hope it is there in the web page, so they will be able to
It is
contact you,
there.
right?
I will tell you because I am so much motivated from Professor M.V. George, because he spent his whole life for the uplifting of science activities in our state. I actually donated part of his his wealth to build the FCBS program. That actually FCBS program has triggered interest among the teachers and as well as the students. If you go to any ISERs or any institute, we can see a number of PhD students, because one way or other way, all the students are somehow connected to FCBS. And even when I was discussing with the Devadas sir, he was telling me that one. So actually, this FCBS program has helped them to start a lot of other initiatives in their department and even research. So I just want to convey that one. If anybody wants any kind of help from our side, because we are part of a CSIR, Council of Scientific and Industrial Research. This is one organization actually started before independence, and this is one of organization will always stand for national interest. So during the COVID-19 time, you can see that one. The maximum number of contributions actually came from CSIR, because CSIR always stand with the society as well as the benefit of the community. So I think as a CSIR, it is my duty to extend any kind of help to my fellow citizens as well as the students, because I know the students are maybe struggling to do some experiments or struggling to have some kind of a help. But anytime they can contact in whatever way CSIR can help them, I think we are doing that one.
Yeah, thanks a lot, Dr. Praveen. Actually, I did my MSc project there in RRL. So I know about. I did with Professor KGK Warrior. And yeah, actually, I came for summer internship there during my first year of MSc. At that time.
Which year you came here? Which year?
Oh, it's long back, 96. So at that time, much before you were joining there.
Just I asked,
probably
you may be
probably your senior, Joby Eldo maybe.
Joby Eldo is my senior. Senior most.
Yeah, he is my classmate.
Joby Eldo is my senior most. Joby Eldo is one of the best researcher I have seen in my life. One of the best systematic researcher.
Yeah, he was a very nice person. He's in US now. Yeah.
When I joined PhD, he was in the final stage of his PhD. Still, we are in touch with him whenever I want to contact him. If you message him through email or Facebook, still we are in contact.
Okay. Yeah, we are all classmates. So I came for summer internship, myself and Srilata. You may be knowing Srilata also then. So we both came there, Joby's wife.
Yeah,
so,
I know.
Yeah, we both came there for summer internship. Directly, they have taken us to Professor George, M.V. George, and he was not at all happy in seeing girls. At that time, the photochemistry division was not allowed for girls. So the reason he was telling is like girls will cry and their long hairs will fall on the floor. So all those reasons he was telling. So yeah, but later he started taking.
Suresh Pilla was before that. He was there 95.
Yeah, Suresh was my senior.
Yeah,
Yes,
like
remote field. Yeah.
Whatever madam is telling that one, this just nearby labs, this KGQRSR labs and
Yes.
photochemistry lab is just opposite.
Right.
Yeah, yeah. Yeah,
So we are
since
studying the
we
same
could
class.
not enter photochemistry lab, we used to go like whenever sir goes to US, he every year, like vacation time, Professor George goes to Notre Dame. So that time we entered to photochemistry lab. So that was the memory with the photochemistry division. And of course, I met him there also. So that time he was telling me, you know why I did not take you? Because girls will cry. So, yeah, now girls are not crying.
No, if you come here in CSIR now most of our students are girls only. There is a very less number of
Yes.
boys are qualifying CSR examination. But it is not a bad thing. What I want to tell you that they are good. That is why they are qualifying examination. There is no question
Yes,
about that.
yes.
They
Yes,
are
yes.
they are very what I want to tell you there. So I have two students from Cochin University. They are very good because it is not like an old time there mainly boys were interested in doing research. Now girls are interested in research. So there is no question
Yes.
about that ability.
Yeah, we can see in
For
the participants also. Many are there. Girls.
if you take an MSc course, for example, if you ask Dr. Sujit, his class, I think 4 or 5 boys were there. In my class, only 2. For my junior, only 1. Out of 16 in our department, only 3 boys were there. So that was the situation. So uh, now girls are more getting attention. That is a good thing. That is
Yeah,
a
that's good.
that is a. I feel girls has a special ability to do multiple number of things. They can do because when I was in Bologna, there was a madam is there, Polo Ceroni. She was just like a mother, like a lady. She had 4 kids. At the same time, she was one of the best photochemists there, and she was HOD also there. She was handling everything. So that is a special kind of ability some people may have. That's okay. I think it's next year science maybe at least in chemistry. I think girls are going to rule if this is the way things.
Yeah, let us hope
A
so.
good thing.
Yeah. Okay, so thank you, Dr. Praveen, for the excellent talk. So we enjoyed a lot. Hopefully, once all these COVID-19 problems are solved, we hope you will come to NIT and we all will have a chance to listen to you directly. Yeah, thanks a lot.
We will meet later.
We
Okay.
will wind up.
Thank you, sir.
Thank you a lot.
Yes.
We can wind up. Okay. Surely we can call Dr. Praveen once to this campus.
Yeah.
Yeah, actually,
Thank you
it will
for
be
your.
really motivation for the students.
Yes, yes,
You
yes,
see.
yes.
So if I get a more time, I will come up with a more result. Latest exciting result. We
Sure,
have
sure.
some other exciting results.
Yes.
Okay.
Okay.
Okay, thank
Then,
you.
madam, we can conclude. Now it is 5:30.
Yeah, yes, yes. So we can
Okay.
leave, right? Yeah, thank you.
Okay, we can.
Okay.
Thank you
Thank you.
all.
Okay, thank you.
Thank you all for the active
Thank you.
participation. Hope all you had a great time. Take the message. Do well. Okay, thank you.
Okay, thank you.
Yeah, bye.
Yes.
Thank you. Bye.